So how did it go? Were you able to open this up in the Numbers app? Anyone not have the Numbers app? Everybody good to go? So this is what a gel would look like after we had 3 restriction enzymes. We labeled those as B, E, and H. BAM-Hi, ECO, RI, and HIN3. And then we had a 4th one right here, the yellow tube in our experiment. It didn't get any enzyme in it. So we would expect that it would just have one big piece of DNA that wasn't cut up. The 3 enzymes are going to cut the DNA in different locations. And remember when we did Who Ate the Cheese and we counted how many base pairs there were in the strand of DNA after it got cut? That's what happens here. So if it got cut with a lot of nucleotides or bases, then it is going to be high up here. So these are the heavier fragments and these are the smaller ones, and they would travel farther. So this is what a DNA fingerprint is. And just a quick review for the quiz. How many different locations on our 46 chromosomes will they cut up and use for DNA fingerprinting? Anyone remember that number? We have 22,000 genes, and you do not do DNA analysis on genes because as humans, our genes are too alike. So when we do DNA analysis, it will show that no 2 people have the same repeating base patterns in what we call non-coding regions. They're not the genes. And they have located how many different spots on our 46 chromosomes that can be used in DNA fingerprinting. Got the number? How many? 13. That's it, right, 13. So only 13 different spots. So they have the restriction enzymes to go after these 13 places, cut it up, and then they'll run it just like this. And we saw this in our Who Ate the Cheese. You match up the banding patterns. We also saw it in Murder at Oldfields, right? So here we're not expecting them to be the same. So we're not doing DNA fingerprinting. We're doing the process of separating DNA out by size. So we put the DNA up in these wells. The DNA is slightly negatively charged, and this is where the negative pole in the gel electrophoresis was. So it's going to push the DNA away from it because like charges repel. Then down here we have the positive pole, and so the DNA being negative is drawn toward the positive pole. So that's what separates it. And then you stain it, and so if you look at your gels over there, they're bluer than that blue liquid because after it ran for a few hours, then I stained it and then spent some time rinsing it. So I was here several hours after school just trying to get your gels processed properly. But they didn't develop, just kind of like in photography. So I think the chemicals were okay. I suspect that the DNA and the restriction enzymes had an issue. So this is what the gel looks like, and we're going to analyze it right now. So the line has already been drawn for you at the bottom, and this is as far as the loading dye that we added to this. This is as far as the loading dye went. And then you shut the gel off so that it doesn't run all the way off the end. If we kept the electricity on here, all these bands would eventually make their way to the finish line. So you want them to be as separated as they should be, and you just don't let it go too far. And now, what we're going to do is we're going to draw lines. It said the first one was done for you. I must have erased it, so we'll do that. Then notice that there's a ruler right here. So this says 13.5 centimeters. So we're going to use the centimeter right here. And then each of these 10 lines between the centimeter is a millimeter. So we could call this out as 135 millimeters. But if we use this as a decimal place between the centimeters, then we can say it's 13.5. So we're going to now draw lines. for each of these. And before we get to it though, notice at the top it's also got another tab that says the gel data table. So once we get that distance from that other page, we're going to write it down here. And then we're going to write it down for each of the 3 different enzymes. And is yours blank or did you have all these lines in it? Yours is blank? Has all the lines? I meant to get rid of these lines, so we're going to use something different. So let's see if we can get rid of them. Yep. So I didn't mean for those to be in there. So take a moment to delete it because this was my practice data. So just click into the lines. And this chart will populate by itself, except for the lines that we're going to put in there. So take a moment to tap into that to get rid of it. I guess I uploaded the incorrect file when I was running through practice on this. So when you go to college and after graduation, realize that spreadsheets are a way of collecting data and analyzing it. You can sort data, you can do mathematical calculations on it. So what I have up here is 3 different tables, one for each of the digestive enzymes. HIN3, ECO, RI, and also BAMHI. If you click over to the 3rd tab on the top right, there are 5 questions for you to answer. The background information in day 1's exercise will help you with a little bit, but let's talk about some of it right here so you have the big picture. Let's go to question number 2. Why did we put the DNA and the enzymes in the incubator? It was at 37 degrees Celsius. Do you know what that equates to in Fahrenheit? What? It is body temperature. It's 98.6. So it's nearly 99 degrees. So this is the way organisms operate best. So what organisms' DNA did we use? It was in the background information. It was one of the questions you had to answer on the day 1. Did we use human DNA? Did we use any other animal DNA? Was it plant? Was it bacteria? Was it a virus? Anybody? What? Right. We used, it was called a bacteriophage. So we used a bacteria, and the bacteria can be infected by a virus. So the virus, when it does its job in all living things, it will inject DNA or RNA. It injects genetic information into an organism. It hijacks the cells, and it takes over control of the nucleus, and then it hijacks everything in the cytoplasm so that the cell will just replicate millions of viruses inside that cell. The only way for it to get out is to be like the Alien movie, and it pops out and busts the cell open. Now you have millions of additional viruses that then go and infect other cells. So when you get infected by a virus, it takes about 2 or 3 days incubation period while this is happening to your cells. You ever feel the chills when you have a virus infection? The chills. You have a fever, yet you're chilly. So when you get the chills, you're reacting to your cells rupturing and releasing toxins from their replication process, and it comes in waves like that. So we used a harmless bacteria, and we used a virus that only infects bacteria, so it wasn't a dangerous thing to be dealing with bacteria and a bacteriophage, a virus that infects a bacterium. We're surrounded by bacteria our whole lives. Did you know that we have more cells of bacteria on us and in us than we do the number of our own human cells? So it's really interesting thought process of taking away all of your human cells and you'd still be in the shape of you from everything that's on the outside of you and inside of you. We have a lot of friendly bacteria and we need them. So what we used was the bacteria's natural defense to kill the virus. So these restriction enzymes that we're using, they're meant to wipe out the virus's DNA that they've injected into them. What a cool defense mechanism they have. So why was the DNA enzyme mixture put into the incubator? Because natural cells, bacteria, and other plant and animal cells operate ideally at a warm temperature. So we wanted the reaction to happen fast. The reason why you put food in the refrigerator is to slow down the action of bacteria so that it doesn't do its job of spoiling the food. If you put it in the freezer, it even slows it down more. So we wanted it to go nice and fast. We incubated it for 20 minutes. Okay, let's go back to the tab on the first one here. It's called Gel, and we're going to follow these directions. We're going to use the color that you see up here. And let's start with the one that's closest. So we're going to use blue. So you'll click on the plus key right over here, and you are going to grab not a table, not a pie chart, but these shapes right there. And you could also do pictures, but we want shapes because inside that shape icon of a circle and a square, there is a line. So tap on the line just once, and it looks like nothing happened, but if you look closely, there is a button right there and one right there, and a black line, which was hard to see on black. So now what you can do is click on the paintbrush up at the top right, and now we can change the color. Keep this, maybe I'll click over here to, this is kind of the thickness of the line, and I'm just going to choose that one. The color doesn't matter because we're going to change the color right there. And we're going to change it to blue. So tap on green and then make it kind of a dark blue in the second column right in the middle there. And now you see that your line is blue. Anyone need help with that? Getting a line? Then make that horizontal. So zoom in on it if it's too small so that it fills the whole screen like I've got. You can follow along here. Yeah. Good. If I got a blue line, and now it says that we want to go to the very bottom of the first one right here. So if I zoom, I can zoom in a little bit. And notice the thickness right here, the width of the line, it's only 1 point. I am going to make it a little bit wider. I think I'll go to 4. It's just easier to see the line. So again, that's under Paintbrush and Style once you've selected on the line. So the paintbrush in numbers and pages is really where you do all your formatting, like you're painting a wall. And so I can click on the paintbrush, it gets rid of that so that you can see it. Select on the line again and tap on it, and then you can click copy. Then you can tap outside of the ruler and tap again and you can paste. I don't know if you can paste right over the table itself, so now what you need to do is do that for the other lines. So once I have that pasted in memory, I just have to, or copied in memory, then I just have to paste it. Then I'm going to do it again, and notice at the top that we need pink. So now I created another line, and I'm going to do the paintbrush. I'm going to go over here to the color and then make it pink. And I'd like the line to be longer, so then I stretched it out, and then we're going to go up here like that. Then I'm going to tap on that and copy it so that I could make 1, 2, 3, 4 more. That third one from the bottom would obliterate the blue line. So then what I did is I just picked it up after the blue line, so I didn't write over the whole thing, because this banding pattern and that banding pattern are the same. How's it coming? We could have done this in Notability maybe a little bit faster if you're not familiar with the menuing system, but we need the database and the charting here. That's why we're not really doing it in Notability. Go ahead and do it for the green column. The third one over. Okay, and now it looks like a little kid has taken a bunch of markers to this, right? So do you have it at the bottom of every one of the bands? And it does snap in place, so maybe we're off a little bit. When we write the numbers down, maybe we can make an adjustment to that. So what we'll do now is take a look at this first one, the blue one. So notice it's between 4 and 5, and it does hit the 7, so it's 4.7 for that first blue line. So now I'm going to use the plus key, and I think maybe I could just start writing on it. Is that the right? Yeah. So I think if you just tap your pencil on the background, do you get this menuing button? Yes? No? Yeah. So we'll use the same color. So go over here and make sure that you have blue turned on. And this first one is 4.7. And then the next one looks like it's 5.2. And then the third one, 6.7. And then the same thing for the green line. So what I think I'll do is I'll just write it over here, 6.7. And the same thing for 7.1. Now I'll switch my pen to pink. And pink isn't in the first choice. You have to hit the rainbow of colors to select your color. And it looks like it's 4.6. 6.3. And this is one where it looks like it's 8.9, but it really need to go up. Am I able to slide that up? Yeah. All right, so yeah, 8.9. Oops. Occasionally, if I tap into this, I'll move the whole thing. Did that just happen to you? I did find that there's a setting here if you're tapped on the, maybe that's just with the chart. So I'll talk to you about it later. If you find that things are moving around on you, like if you were on the picture and all the lines stayed the same and now you're moving the whole picture, then pinch it in so that you could tap outside of the picture and then maybe tap into it again, slide it around, make some adjustments. I'll come around and see how you're doing. Let me fill in the last one right here. And so I need to change my color to green. And this is 4.4, 5.7, 6.7. That was 7.9. Excuse me?
Is it okay for a little off, or should we just copy yours?
You can go off of your numbers. Yeah, that's fine. You're not going to be too far off. Notice we've got colors right here, and to shapes. And it's right here, really small. And the shape's the size of 4 copy it, then paste additional ones, because you need is like as many as you see here in this column. So you need 4 green lines, something like 6 pink lines, and so on. Then we draw lines right underneath the banding pattern, and using the ruler where it intersects, we're writing down how far that band moved in the gel. So you can catch up with us later. This is recorded too, so if you need help, I'll help you a little bit. But you kind of got caught up. Get on the line? Right, yeah. Kind of works to write them in white. That's not a bad idea. All right, now all this data is going to go over to the next tab. And it's not ideal, right? Because you got to remember the numbers or write them down somewhere. You can take a picture with your phone, then you can deal off the picture on your phone. So that's kind of the easiest way instead of having to write it all down again. So if you'd like to take your phone out, take a picture of it. And then as I go over here, now we're going to write down the distance that we had in our gel. So now it's pretty easy because it's all color coded. And so the first one that I had was 4.4, 5.7, 6.7, 7.9, 11.0, 11.8. And if those are your numbers, great. If they're a little bit different, and why do I have a line around that one there? And do that for distance migrated for all the other colors here. So now pink, I had 4.6, 6.3, 7. Oh, and now here is where, notice that the actual size is only 200 base pairs apart and the gel cannot separate them. They actually go over each other. So for my third one here in pink, it's 7.6, and you have to type 7.6 for the second one. So all the duplicates I highlighted in yellow. These are duplicates, and those when we get over to BAM High, but there were no duplicates over here. They were far enough where they would be individual bands, but these bands overlap. And then my last two bands, 8.9. Oh, looks like I missed one. The duplicates are 7.1. And then green. There's 2 of those. Uh-oh. I just realized I was reading them in the wrong order. Were you guys, nobody caught that? So green is actually, I'm looking at the wrong color. So let's see, 4.4, 5.6. Okay, I did it right. For the last one, I wasn't reading blue. 4.7, 5.2, 6.7, 6.7, yep. And then the last one, 7.1. So if you're following along with my numbers, that's what I have. So these duplicates, make sure that in the yellow bands and in this one, you realize that they're only 200 base pairs apart, and this is 600. Still, they overlap. And this one was 300 apart base pairs, and that's 100 base pairs. So those bands overlap. They don't get separated with these restriction enzymes. Now, what you see down here in the chart is that some numbers popped up. And they're pretty much in a straight line, but we can't connect them by a curve. And we're not going to connect them by straight lines, otherwise it would be straight line, then go down, then go this way. So we're going to do what's called a best fit line. And I think you've done that in math before, right? Software programs can calculate it, the best fit line for you. We're going to eyeball it. So we'll try to have as many on the line as possible. Then if we have 2 up above the line, we'll try to have 2 below the line. And we want to try to eyeball it so that they're equidistant from the middle of the line. So we are going to make a best fit line with HIND3 enzyme. So now notice there's a column that says RF. In chemistry class, you did this with your chromatography. That's the relative frequency of movement from the gel to as far as the separation went. That was the line that we made right here. So this line at 13.5, that's as far as our front went. So we're going a relationship between the gel and as far as the whole gel ran, called the front. The RF value is in chromatography, separation. So if you notice now, it has done that for you. It calculated it automatically with the information that we put in right there. So if the front was 12, it would change all the RF values because all these RF columns are already built in formulas for you to make this go a little bit easier. But notice we have 2 more columns to fill in right here. It's called interpolation. So we're going to use a technique of estimating based on a standard. So our standard is HIN3 over here with the best fit line. So let's do our best fit line. Let's make a green line. It's probably still in memory, one line or another. And the last one I did, I just pasted and a green line popped up. If you have a different color line, then you just go under the paintbrush and then change the color. So now you have a line that we're going to tap into and angle it, stretch it out, and we are going to be doing these lines like that. They do say that this guy right here is an outlier. Do you know what outliers mean when you're doing average? It means that it throws off the curve or it throws off our best fit line. So we are instructed not to use that one because it is a huge base pair. So notice we have 2,000 and 9,000 and then it jumps all the way up to 23,000. So if we try to include that one, it's going to skew all of our other ones. So you should try to do a best fit line just based on the others. So how does it look? There is some variation in doing this unless you were using some sort of calculator. And I think Excel can do a best fit line, but numbers can't do it. So I'm going to eyeball that. You know, there is some variation here, so maybe you think that the best fit line is like right there. That looks pretty good because I have that dot right there, and then these two are above and those two are above. Oh, I have too many above. So I would want to have some below the line, so that wouldn't be a best fit line. It would be more like two are hitting it, one's below, one's above. Maybe that's the best fit line. I'm going to go with that. All right, we don't have too much left to do. So we're going to take a look at EcoRI, and we'll start from the RF of 0.34. So go down here, remember that 0.34. Grab another line, paste another line in here, and make it pink. All of our lines for this next one, pink or purple, it needs to be a vertical line. Have it go 0.34. So what you see here is every dashed line is right in between the two that are labeled. So 0.35 would be that dashed line, and 0.34 is just a tick to the left of it. This is not a standard chart. Look at the y-axis. It goes from 1, 2, 3, 4, up to 10. And then it goes 10 to 100 with 10 divisions in there. Then it goes 100 to 1,000 with 10 divisions. Then 1,000 to 10,000. Anyone know what you call this when it continues to change the y-axis? It's not numeric. It's called, starts with an L. Logarithmic, a log chart. So this is a way of showing data that is not linear. But it is in a sense because it goes from 1 and then every division is 1. But look at how wide it is at the bottom, and then it gets smaller and smaller, and then it's wide and then it's smaller and smaller. So it keeps it a steady increase in the y. So it's really interesting, isn't it? Dealing with a logarithmic graph. So what we're going to do now is realize that every dashed line is 10 between the two marked. So here we are. I zoomed in on where it crosses our best fit line. And this is 10,000. And this right here is, it's between 10,000 and 100,000. Each dashed line now is another 10,000, so that's 20,000. So if we're right here, 1, 2, 3, 4, 5, 6, 7, 8. What was that? Just about. I like to eyeball it, half and then half. So it's just a tick above 10,000, right? So I would call this 20,000 right there. Does everybody kind of get that? If you would like to make another line for yourself, but I want to have pink lines, so I'm going to copy that into memory so that I don't have to keep changing the color. So even though I'm slightly above 10,000, I'm almost at 20,000. Nope, that's 20,000 right there. So it's 1,000 above that. 10,000, 20,000, 30,000, 40,000, so 20,000. So I would call that 11,000 or thereabouts. Open to interpretation. So now I'm going to go up here and I'm going to say that my interpretation is that that's 11,000. The reason why we're so far off of what it actually was is because our logarithmic chart cannot do a high number. We left that guy out. But we're going to write it in there anyway. It doesn't fit our best fit line, and we knew that because it's too high of a number. See how you do on the next one. I'll do one more with you, and then go ahead and work ahead of me so that you get all of them. I'm not going to do all these for you. I'm just going to get you started with it. So our next RF value is 0.47. So I'll paste another line, make it vertical, and go to 0.47. Point 4 to 0.5, that's 0.45, so 0.457 should be a little bit farther to the right. You want that line to cross the best fit line? And it's okay if you don't want to draw the horizontal lines because you have these dashed lines which allow you to interpolate where it's going to cross the y-axis. So it was right there on this dashed line. I'll just follow it all the way across. And it's right here between 1,000 and 10,000. Every one is 1,000. So that's 1, 2, 3, 4, 5, 6, 7,000. 7,000 base pairs. So what I'm going to do now is write in the color that was 7,000. I think we called this 11,000. And these are BPs or base pairs if you look at the y-axis. So we calculated it as 7,000 when in actuality, if I didn't give you these numbers and then I graded you on it, I would like to see that you get close. So look, we got 400 base pairs. That's really good. I'll do one over here in the blue, 0.47. It hit the same, no, the RF value, 0.35. So now I'm going to do this one in blue, 0.35. It's going to go right over the dashed line. And it should be a little lower than the pink line to the left of it. So I think I'll call that 10,500.
Where are you getting the base?
The y-axis is base pairs. So I'm looking at where that vertical line hits the best fit line, and then you draw a horizontal line all the way over to the y-axis, and you read it right there. So 10,500. It'll have a little bit of trouble with that one because that one was above our best fit line too. but all these other ones are going to be pretty close to what it is. So let's check. I'm going to check out one more. 0.39. Okay, so you have 1, 2, 3, 4, 5 more to go from what I did. Answer the conclusion questions, and then you hit the 3 dots. Export this as a PDF file and fit each sheet to a single page. Send it over, export it over to Notability. Then if you opened up Notability, you'll see that you have a page for each one, a page for each tab, and that's your write-up. So yeah, I went a little fast, and if you've never done this before, you know there's a learning curve. So I will help you with this as we go through the rest of the class. So we have 25 minutes. Should be enough time for you to handle that, and then turn it in. Then your homework is to get ready for the quiz, or it's almost like a quiz. It's 40 points, and I think it's less than that many questions. So it goes over the basics of this lab right here, understanding that we have a bacteria's DNA and we're using, no, it's the virus's DNA and the bacteria's restriction enzymes, which cut the DNA up into these pieces that we see on the gel. Then we did the work of a scientist in calculating how many base pairs is that off of a standard. So our standard was the green right here, this big long one in green, hint 3.